In this video, I wanted to talk a little bit about the electron transport chain. I know that this can be a daunting process with a number of moving pieces, so I wanted to try and make sure that we are framing electron transport in the lens of energy transfer. Right? Because remember, the ultimate goal of cell respiration is to take the chemical energy that's stored in the bonds of a molecule of glucose and transform that energy into the chemical energy in the bonds of a molecule of ATP that we can use to power our cellular processes. Now, to this point, we've already done glycolysis and we've done the Krebs cycle. We've taken that molecule of glucose and we've completely ripped it apart into 6 carbon dioxides. Energy-wise, carbon dioxide is a pretty dead molecule. We cannot get any more energy out of those molecules of carbon dioxide. So those can go to our lungs and get out of our body. After glycolysis and Krebs cycle, the energy that was in that glucose molecule is now mostly stored in our high-energy electron carriers. We've made all of these molecules of NADH. We've actually made a molecule of FADH too, but for this class, we don't need to worry about this. It works in the same way that NADH does. So on principle, we can consider this to be the same. You'll learn about the difference between FADH and NADH if you move on in biology, but for this class, let's just consider all energy carriers or all electron carriers to be the same. If you'll remember, we've also made a couple of molecules of ATP through glycolysis and Krebs, but those are not going to be ultimately that important. So the majority of our energy in glucose is now stored in the bonds of these electron carriers, right? These are the high energy form. They have an electron. How do we get the energy from these electron carriers into ATP? That's the question we need to answer with the electron transport chain. So we're going to set ourselves up here. We are looking at the mitochondria. So if you remember, glycolysis happens in the cytoplasm, and our molecules of pyruvate are going to move from the cytoplasm into the mitochondrial matrix, right? This is the inner space in the mitochondria. The matrix is where the Krebs cycle is going to occur, and most of our NADH is going to get produced. The machinery for the electron transport chain is located in the inner membrane of the mitochondria. So you can see that all set up right here in this inner membrane. If you remember, the mitochondria is a double membrane organelle, which means that there's an inner membrane and outer membrane. We see the outer membrane up top here. In between them, we have what's known as the intermembrane space. This is a space between the two membranes, and this is going to become important in a couple of slides. So what we have here is our electron transport machinery set up at the inner membrane of the mitochondria. What electron transport is we have this setup of electron carrier molecules. So much like our NADH, right, these electron carrier molecules are going to take an electron from one molecule and then bring it to another. What happens at the start is our electron carrier, our NADH, our high energy electron carrier, is going to come up to the first carrier molecule in this chain. This carrier molecule has a slightly higher redox potential. Now what that means is that this molecule wants the electron a little bit more than NADH does. So what's going to happen is NADH is going to transfer the electron onto this molecule. The NADH is now in its oxidized form, this NAD, the low energy form. This can go back out and grab another electron from a molecule of glucose and start the process again. But we're thinking about this electron here. So this electron gets transferred to this carrier. This carrier here has a slightly higher affinity for the electron than this carrier does. So this carrier transfers it here. This carrier is going to transfer it here, and so on, because each of these subsequent carriers wants the electron just a little bit more than the carrier ahead of it. Now, since we have a slightly higher potential, as we transfer the electron, it's going to be more and more comfortable on these molecules, and that means that its transfer is going to release a little bit of energy. Right? So with each subsequent transfer, we release a little bit of energy. And we're going to talk about what happens with that energy coming up. So what we can see is all of these different proteins are shown on this chart. They're color-coded here, but we have an increasingly positive potential, which means that the electron is going to flow in this direction. So don't worry about the math of this, but the electron flows in this direction, and as it does, it's going to release a little bit of energy with each transfer. Now, ultimately, our terminal or our final electron acceptor is our molecule of oxygen. So we mentioned that between glycolysis and Krebs cycle, we've never mentioned oxygen before, even though we're doing aerobic respiration. Here's where the oxygen comes in. The oxygen sits at the end of this electron transport chain and is ready to take those electrons off the chain and become water. So oxygen, when it accepts electrons, it's going to turn into water. Don't worry about how that works chemically, but just know that the oxygen that's serving as a reactant in cellular respiration is going to grab electrons and then turn into the water that's coming off as a product. So this is the stage where we use oxygen and we generate the water, and that's the process of dropping these electrons onto a molecule of oxygen. So we mentioned that each transfer of the electron is going to release a little bit of energy. What do we do with that energy? The answer to that is we are going to start moving protons across the membrane. So protons, H+, are they are a particle within the mitochondria. Well, within all of our cells, they're related to pH. But what a proton is not really here or there for this discussion. But they're going to move protons. They're going to move them against their concentration gradient. And what that means is that there's going to be a lot of protons in this intermembrane space, and not a lot in the mitochondrial matrix. Now, the fact that we have a lot of this particle in this little space up here, we have a high concentration up here and a low concentration down here. Right? Nature does not like when there is a separation, when there is this gradient, when there's a lot of something in one place and not a lot in another place. Nature likes things to be even. So the natural tendency of things is to move around and even each other out. So to create and maintain this disparity, this high concentration of protons in the intermembrane space and the low concentration of protons in the matrix, this takes energy, right? So right now, the energy here is stored in this concentration gradient. This is a form of potential energy. So we drag these protons up into the intermembrane space, right? We're using the energy from electron transfer to drag these protons into the intermembrane space. And the fact that we have a high concentration in here and a low concentration in the inside the mitochondria is a form of potential energy. Now, what's going to happen is that potential energy is going to want to come back to zero, right? These protons are not they don't like being all jammed up in the intermembrane space. They want to fall back through the side, back into the mitochondria. Now, to do that, they really can't pass through this membrane because these are positively charged, and these lipids don't like interacting with positively charged ions. So in order to come back down this concentration gradient, right, to release this energy that's stored in this gradient, these protons are going to fall through a molecule of ATP synthase. This is the enzyme that is going to create our ATP. As protons fall down their concentration gradient, they are going to release energy. Right? They're going to release energy into this molecule. This molecule is going to spin around, and that spinning is going to be used to attach a phosphate onto ADP and make our ATP. Right? So as the protons fall down their concentration gradient, they're converting this potential energy stored in the gradient into kinetic energy, them moving across the membrane, and this energy is then converted into the chemical energy of ATP. So at this point, we have taken our protons, we've let them fall back down the concentration gradient through the ATP synthase, and the energy released as the protons fall back into the matrix is going to be used to phosphorylate ADP and make our ATP. This is where we make the brunt of our ATP. We transfer the chemical energy in NADH, and then we transfer the chemical energy stored in the electron of NADH into these electron carriers. As we move down this electron transport chain, we are going to use the energy that comes off from these transfers to pump protons into the intermembrane space against their concentration gradient, creating this potential energy. When this potential energy is released by protons falling back down into the matrix, that release of energy is harnessed in phosphorylating this molecule of ADP to make our ATP. So and here's the ATP synthase protein. As the protons fall through, this protein is going to spin around and use that spinning energy to make a molecule of ATP, or to phosphorylate your ADP into ATP. So, to summarize the process of cellular respiration, we start with glucose. Through the process of glycolysis, we're going to split that up and make 2 pyruvates. We're also going to make a couple of these high energy electron carriers, as well as a couple of ATP. We move the pyruvates into the mitochondria, where we then convert it to acetyl-CoA and release a couple more ATP. The acetyl-CoA is in the Krebs cycle, and as it spins around the Krebs cycle, we're going to release a couple of ATP, as well as some of these high energy electron carriers. The electron carriers from glycolysis, this linking reaction right here, as we get into the Krebs cycle, and these energy carriers here are all going to go to the electron transport chain. So we talked about glycolysis and Krebs cycle producing some energy. We get 2 ATPs from each. The electron transport chain releases 32 or 34 ATP, right? So the ETC is going to release about 9 to 10 times more energy than glycolysis and the Krebs cycle. So all told, for a molecule of glucose, we get somewhere between 36 to 38 ATPs. We've taken the chemical energy in our glucose and we've converted it to the chemical energy in ATP.